Hi, Assalamualaikum warahmatullahi wabarakatuh. So today we are going to learn about muscle contraction. This is presented by Group 2. These are the presenters. First we have Aida, and next we have Patricia, next we have Christina, and me, which is here. So these are the learning outcome. First is to differentiate type of muscle tissue. Next is to identify microscopic anatomy and physiology. And to describe the mechanism of muscles contraction. Basically, you need to learn how muscles contract, and you will learn about muscle will pull, not push. Alright. So these are the muscles in our body. Muscles is important to do our routine every single day. Okay. So muscles is composed of contractile tissue capable of changing its length by contracting and relaxing. Basically, our muscle contract and relax, and it's very dense with sound. So most animals rely on muscle for movement. Everything, cat wanna walk, fish swim, bird fly. Everything need muscles to move with. Okay, only a few aquatic animals does not rely on muscles, that they are non-motile, which is they eat current, so they don't need to use muscles for movement to get food. So there are three distinct types of muscle tissue. First is smooth muscles, second is cardiac muscles, and third is skeletal muscles. Smooth muscles is involuntary muscle that show no cross-stripe under microscopic magnification. So involuntary muscles mean you don't need to think about what you want to do. Okay, so involuntary muscle, for example, like perithalsis in esophagus. Next is cardiac muscles, also called heart muscles or myocardium. This is the foundation of heart. And next we have skeletal muscle. We're going to learn this in the next slide. So skeletal muscles, so microscopic anatomy and physiology. So skeletal muscles attached to the skeleton by band of fibrous connective tissue called tendon. So basically, muscles are connected to skeleton by tendon. Alright, so it pull not push, like I said, it's arranged in antagonistic pair, which means, if you can see my screen, my camera. Okay, if you want to grab something on green, right? If you want to drink, my bicep will contract and my tricep will relax. And if I want to take my arm, my bicep will relax and my tricep will contract. So that's mean antagonistic pair, which is one of muscle will flex the joint and bend limbs, and the other joint is straightened to the limb, right? So muscles at rest exhibit minimal contraction, which is when muscle at rest, muscles exhibit tone when at rest. Tone means that our muscles contract partially, alright? Because when they are completely relaxed, we cannot stand at all because no contraction happen in muscle. So we need tone to maintaining our posture. So muscle sphincter and Golgi tendon are the responsibles for this maintaining tone.
Next is the microscopic anatomy and physiology. Each muscle fiber is a cell containing the cellular components, but some components have special features. First, we have sarcolemma, which is a plasma membrane that forms a transverse system called T-system. The T-tubules penetrate to come in contact with sarcoplasmic reticulum. Next is the sarcoplasmic reticulum. Sarcoplasmic reticulum consists of modified smooth endoplasmic reticulum. This reticulum able to store calcium ions, which is essential for muscle contraction. The sarcoplasmic reticulum encases thousands of myofibrils, which are the contractile structure in the sarcoplasm. We can see from the picture that the myofibrils are cylindrical and have length of muscle fiber. Myofibrils contain light and dark bands under light microscope. The function of these bands are for skeletal muscle striated appearance. The last special features is the sarcomere or the protein filament, which arrange linearly within a myofibril. Sarcomere is the units of contraction within a myofibril, and they are composed of two protein filaments, which are the myosin filament, also known as the thick filament, and the actin filament, also known as the thin filament. This is the sarcomere. When sarcomere relaxes, it extends between two dark lines called the Z lines. We have two regions of band. First is the light region, which is called the I band because it contains only active filament which attach to the Z line. Next is the A band, also known as the dark region because it contains overlapping of actin and myosin filaments. The last one we have H zone, which only contains myosin filaments. Now I will explain about the sliding filament model. This is a sarcomere and this is another sarcomere. The green filament is the active filament and the purple filament is the myosin filament. The active filaments are found at both ends of the sarcomere. You can see the active filaments are found at the both ends of the sarcomere. One end of each filament is attached to a Z line at one end of the sarcomere. One end of the active filament is attached to the Z line, while the other is suspended in the cytoplasm. While myosin filaments are suspended between the Z line. So from here you can see how the active filament move towards the myosin filaments. This is how our muscle contracts. Now I will explain how the sliding filament model works. When a muscle fiber contracts, active filaments slide past the myosin filaments and approach one another. You can see here the active filaments move towards each other and approach myosin. The sarcomere shorten causes the Z lines to move towards each other. However, during the sliding process, the sarcomere shorten even though the filaments remain the same. For your information, the movement of active filaments toward the myosin filaments are called the sliding filament model of muscle contraction. Now I will pass the presentation to Batisha.
Hello everyone. So my name is Nur Badisha and I will continue our presentation with working muscle require ATP. So from the G, we can see that myosin filaments will break down the ATP to form the cross bridge with actin filaments. So the actin filaments will pull toward the sarcomere. So the muscle will contract. And sustained exercise requires cellular respiration to regenerate ATP, so we can use the ATP again and again to contract the muscle. Next, we move to the use of ATP in contraction. Although our muscle have myoglobin, which is a molecule that contains oxygen, cellular respiration doesn't immediately supply all of the ATP that is needed. Meantime, the muscle fibers rely on creatine phosphate. Creatine phosphate is actually a storage form of high energy phosphate. But this creatine phosphate cannot directly participate in muscle contraction. So therefore, it needs to anaerobically regenerate ATP. By this process, where creatine phosphate with ADP to become ATP plus creatine. When creatine phosphate is depleted and the cell still needs more ATP for muscle contraction, it will undergo fermentation, which is a process that not consume any oxygen. So when fermentation occurs, it will supply ATP for a short time when we do strenuous exercise. But the bad side of the fermentation is it will build up a lactate that causes the muscle to fatigue. Lastly, the continued intake of oxygen restores cells to their original energy state, offsets oxygen debt, and the lactate will transport to the liver. Next, we move to the function of myosin. So the first picture over here, you can see this is actin filament and the myosin head. When ATP binds with myosin head, the ATP will split into ADP and phosphate. And the breakdown of ATP on the myosin, it allows the myosin head to attach to the actin filament to form cross bridge. When the cross bridge is formed, ADP and phosphate will release and the cross bridge will change its position and the power stroke occurs. that pull the actin filament toward the center of the sarcomere. And the ATP binds to myosin head. The cross bridge will broken and heads will detach from the actin filament. And this is where it completes one cycle of contraction. After breaking down ATP, myosin head binds to an actin filament and later a pulsar causes the actin filaments to move. Now when another ATP binds to myosin, the head detached from actin and the cycle begins again with the presence of the calcium ion. So the cycle will repeat and actin filament will move closer to the center of the sarcomere. That's all from me. Thank you. To the next presenter.
Assalamualaikum and selamat sejahtera to all my friends, K19 and my lecturer, Miss Atikah. So my name is Aida, your presenter for today, and in my part, I'm going to explain about the calcium's role in a muscle contraction. So the main point here, calcium's function in a muscle contraction is actually to bind with the troponin to block the active site of a myosin. Okay, I know you guys are confused, but let's go deeper into the flowchart that I made for you guys. Okay, so the first step here is, okay, before I explain about the first step, I want to remind you guys that the first, second, and the third step happen in a very short time. So it's very fast. Okay, let's look into the first step. So the first step here is acetylcholine will bind to receptor in the sarcolemma. If we look closer into the diagram, acetylcholine is actually one of the neurotransmitter that will be undergoes the synapsis process and binds to the receptor in the sarcolemma. And if, okay, so the neurotransmitter will be changed into the impulse signal that will spread down to the T tubules of the sarcoplasmic reticulum. So we can see that here, after the acetylcholine goes to the sarcolemma will pass the impulse from the sarcolemma into the sarcoplasmic reticulum. So after the sarcoplasmic reticulum receives the signal, it will release a calcium. Okay, the calcium is released from the sarcoplasmic reticulum after sarcoplasmic reticulum receive the signals. I mean the impulse from the sarcolemma. Okay, let's look into the next step. Okay, the calcium that we got just now will be used, will be bind with the troponin. Here, if we look into the diagram, we can see the troponin structure, the orange one here, and it will regulate the position of tropomyosin that will block myosin binding sites on actin filament. Okay, if we look closer into these two diagrams, we can see that the position of a troponin here and the orange and the blue line between these two diagrams is different. We can see that here the orange, the troponin is in the top, and the second diagram, the troponin is at the middle. So we can see that there's a change in the position of the troponin, and also there's a change in the tropomyosin that will block the actin filament from binding with the active sites for myosin. Okay, the next step is, so the myosin binding sites are exposed, and the myosin head start a binding process, thus forming a cross bridge. We will look into the diagram here, and we will see that the myosin head will be bind with the myosin binding site. Here, the dark green one, the dark green hole here, this is the myosin binding site, and it will form a cross bridge. Okay, the next step is the power stroke pulls thin filament to middle of sarcomere. Okay, we can see that if they form a cross bridge, they will pull the thin filament into the middle of the sarcomere, which is the shortening of the sarcomere is happening. That will lead to a muscle contraction. This is how muscle contraction can happen in our daily life. Okay, let's go to the next part. So the next step is a cross bridge are broken when the head detached from the actin. This can be happen if the new ATP bind to the head, resulting in the detachment. So we can see here in the diagram that the myosin head is no longer attached to the myosin binding site here, and this will result in a muscle relaxation, and the muscle will not be contracting. So the muscle will be relaxing, and the cycle will repeat it over and over again to make a movement, which is a muscle contraction. Okay, let's recall what we just learned from my part. Okay, so the first one is acetylcholine, which is a neurotransmitter, binds to receptor in the sarcolemma here. And the second step is the sarcolemma generates impulses that spread down T tubules to the sarcoplasmic reticulum. And the sarcoplasmic reticulum will release a calcium. So the calcium will be used to bind with a troponin that will regulate the position of tropomyosin, which can block myosin binding sites on actin filament. Here. So The next one is myosin binding sites are exposed. The myosin head start a binding process, thus forming a cross bridge. So the purple one here is a myosin head and attached to the myosin binding site that will lead of reforming a cross bridge. So this cross bridge will generate a power stroke pulse in filament to middle of the sarcomere, which is the shortening of the sarcomere. If you can see here, there is a shortening of the sarcomere, which is lead to the muscle contraction. Okay, and the cross bridge are broken when the head detached from the actin. If we want to detach the head from the binding site, the new ATP must be bind to the head, resulting that will lead to the detachment process. So this cycle will be repeated over and over again to make a movement, which is a muscle contraction. So I hope you guys can process this flowchart very well. Okay, so here's some video link, a YouTube video link that I provide for you guys. If my explanation is not good enough for you to understand well, you can go and look at this video, because this video is very helpful explaining in this part. That's all from me. Thank you.
So guys, we come to our summary. So what we learned in this presentation, first type of muscle. There are three types of muscle that we learned is skeletal muscle, and next is macroscopic anatomy and physiology. The anatomic pairs, and next is sliding filament model, and also calcium in muscle contraction. What calcium do in our muscle contraction? So these are the references of today's presentation. So thank you for listening. So don't hesitate to ask any question. So thank you, that's all from us.